This is the Surgery and Neuroscience of Addiction chapter. Um, I am Michael Caplet, professor of neurological surgery, vice chair for research and residency program director in the Department of Neurosurgery at Weill Cornell Medical College. And I am recording this on behalf of myself and my co-author, Santiago Unda, MD, a postdoctoral fellow in our department. Um, Now, addiction, as most of us know, is a harmful, compulsive, physical or psychological need that cannot be controlled and or causes symptoms upon withdrawal. So this can be a physical dependence, such as substance abuse. This can be a psychological dependence, etc. And so there are many types of addictions, and the chapter that we have reviewed focused primarily on drugs of abuse, and in particular opiates and cocaine, because the neurobiology of this is among the best understood. But there are many different types of addictions. There are the obvious drugs of abuse: cocaine, heroin, methamphetamine, many others. There are what we refer to as the legal social substances, things that are legal for people to take but still have high addictive potential. These include alcohol, nicotine, and cannabis is in question marks because it has increasingly been legalized in many places throughout the United States, and likely will continue to do so. So it will be very much akin to alcohol and nicotine in terms of how it is being used and its availability, but also has clear addiction potential. One of the interesting things about cannabis that is not discussed much in the political discussions is that cannabis itself has a modest addiction potential compared to the more severe drugs, but there is good data that there are higher rates of use and abuse and addiction to the more severe drugs of cocaine, heroin, and others when people use cannabis regularly. Then, of course, there is the abuse and addiction to prescription drugs, and most people are aware of the severity of the opiate problem in the United States and elsewhere. That has been the subject of much discussion in the media. There can be addictions to non-prescription drugs of many types: stimulants, drugs, allergy drugs, etc. And then there can be behavioral addictions, addictions to gambling, addictions to sexual behavior, social media addictions, many others. And these can be as devastating or more devastating on patients. And most of these addictions, even though the mechanism of addiction may vary in terms of specifically how these different things lead to a dependence or an addiction, ultimately much of the circuitry that regulates addiction is very common to many of these different areas, and that presents opportunities potentially for neurosurgeons to intervene, because neurosurgeons obviously can be helpful when we have clear anatomic targets that can be modulated using surgical tools or implants that have been shown to be effective in other areas of neurosurgery and neurology. Now, the epidemiology, we all know that this is a big problem. A 2019 survey showed that 60% of U.S. adults had used some substance with abuse potential within 1 month of answering that survey. Over 20 million U.S. adults are addicted to drugs or alcohol. Long-term opiate dependence affects between 1.5 and 2.5 million people. Now, what is interesting about this statistic is that it seemed to have peaked around 2016 during the worst of the opiate epidemic. And by 2019, it was reduced. So 2016, it was around 2.4 million people were estimated to be addicted to opiates, to prescription opiates. And it was down to about 1.6 million by 2019, likely due to changes in prescription patterns, greater awareness of the problem, and in all likelihood, greater caution among practitioners because of the concerns about the political and legal issues, as well as the addiction issues surrounding this problem. Cocaine dependence affects 2 million people, and half of those people meet criteria for cocaine use disorder, which is the more serious addictive component of cocaine abuse. Now, for all of these things, alcohol addiction, opiate addiction, cocaine addiction, and many others, there are very few approved therapies. The mainstay of treatment, of course, is psychotherapy, 12-step programs, inpatient rehabilitation therapy programs, ongoing support systems, buddy systems, etc. These have variable success depending on the individual and the nature of the addiction. In terms of more direct pharmacological intervention, there are very, very few options for practitioners. There is methadone, of course, for opiates, but methadone is essentially replacing one more severe addictive substance, which are prescription opiates, heroin, narcotics, etc., with a presumably less severely disabling substance that they remain somewhat dependent on. Methadone is something that patients continue to take for years, if not indefinitely, in clinics where it is provided in a controlled fashion so that they don't overuse it, but they are still relatively dependent on the methadone. So this is not like, for example, nicotine gums or other things that are intended for short-term use to try to break the dependence on nicotine, and those also have variable success in terms of recidivism. But that is the goal of methadone, is essentially replacing a lower abuse potential drug that can be managed at clinics for a higher abuse potential drug long term. There's naltrexone, which is an opiate inhibitor. It is often used in emergency rooms for toxicity, but it can also be used in the right way in a controlled fashion for treatment of opiate addiction. But that is really it. There are no FDA-approved therapies for cocaine addiction and for many other addictions. And practitioners use a variety of off-label medications, often involving the dopamine system that we will talk about in a few moments, to try to address this. And so it is clear that there are important unmet needs that could be addressed by the neurosurgeon. One of the values of functional neurosurgery is that we can target brain regions specifically without affecting the rest of the brain. And the problem with addiction is that most of the attempts at pharmacologically treating addiction are somewhat limited because of the profound effects on brain regions outside of the circuits that regulate addiction that can have very strong adverse effects. And we'll talk about that in a moment. So how do we understand the circuitry? How do we break down the circuitry of addiction? Well, there are many tools that have been used over the years, but the most exciting tools in recent years have been those that allow us to take control exogenously of specific populations of neurons and turn them on and off. The most well-known of these techniques is called optogenetics. The idea is that you have a modified ion channel that were derived from ion channels in algae and other organisms that live deep in the ocean, and that need to identify very dim sources of light. And so they can move towards sources of light by having ion channels that activate neurons in the presence of light. So these were identified, cloned, and then could be put back into neurons in mammals, in rodents and other mammals. And in fact, in the presence of a specific wavelength of light that channel reacts to, these channels would activate. And so you could have channels that would allow sodium to go in, and that would depolarize the neuron. You could have channels or pumps that would allow chloride to go across, and that would hyperpolarize and inhibit the neuron. And so in response to the specific wavelength of light that activates that channel or pump, you could then depolarize or hyperpolarize neurons that expressed that channel. So you would put a light fiber into the brain, and that in the animals, and they would move around with the light fiber. And then when you turn that on, they would have a particular behavior. And with that, very specific circuits have been identified. Another major advance in this have been so-called Cree driver mice, where you have mice that express an enzyme called the Cree recombinase, which is a bacterial enzyme, in specific cell types by using very specific promoters that will allow expression only in cells that normally produce that gene. So if you have a promoter, for example, for one type of dopamine receptor, let's say the D1 dopamine receptor, if you use that promoter to drive Cree expression, then it will only express in that transgenic animal in D1 containing neurons. And so if you then put in a virus, a gene therapy agent, into those animals, into a specific brain region that you're interested in, and the so-called opsin, the ion channel gene, is inactivated normally by a sequence that could then be excised in the presence of this Cree recombinase enzyme, then only those cells that express Cree will allow activation of the opsin. And so when you put that virus into a so-called Cree driver mouse, only the cell types expressing Cree will allow that ion channel to be expressed, and then it will only influence the neuron when the light is turned on or off. So you can get very exquisite spatial, anatomic, as well as temporal control of neurons turning on and off. And with this, a variety, particularly in the psychiatric field and particularly in addiction, a variety of circuits have been identified through this. Other techniques that have now come along are analogous but allow different ways of approaching this. One is called chemogenetics. As the name implies, a drug will allow an ion channel to open or close in response to a particular drug, or will activate a second messenger system in a cell. And again, the drug can then activate or inactivate certain neurons, and you can then use surgery to deliver this to specific brain regions, use Cree driver mice to deliver this to specific cell types within that brain region, and again, try to dissect things out. But the temporal nature of chemogenetics means that the behavior is not as immediate, but it is it can be done more chronically without tethering to a device. And then there's a newer technique called magnetogenetics, where you can use magnetic fields to do the same thing using a modified so-called TRPV1 receptor that is attached to a ferritin nanoparticle, so that in the presence of the magnetic field, this ferritin nanoparticle that has iron molecules in it will essentially open the ion channel, achieving the same goal, so that you can, without a light device, open and close these ion channels. So these are among the techniques that are being used to dissect the neurobiology of addiction. And then the animal models that are used to test some of these hypotheses can be varied. Obviously, there are limitations in what one can do in animals that don't have the complexity of the human brain, but the reward circuitry that seems to mediate addiction is a very primitive circuitry that seems to be more analogous from rodents up to higher animals than many other circuits that influence cognitive or behavioral functions in human beings. So for cocaine, there are three different types of models. There is the self-administration model, where an intravenous is put in and tunneled under the skin, and it is attached to an infusion system that's attached to a lever, where the animal can push and self-administer the drug in response to a variety of cues, etc. And the more they and presumably over time, they get used to self-administering more and more, and that can be quantified. Another measure of addiction is locomotor activity, or so-called locomotor sensitization, where the animals will move more aggressively and to a greater degree in response to cocaine, and that can allow you to quantify the cocaine effects on the animal. And then finally, there's conditioned place preference, where the animal is given cocaine in a particular environment in a cage, and there are two sides or more sides to a given cage, where there are some cues that tell the animal which side they're on. And then you put the animal on a particular area that has those cues, and they get the cocaine each time, and then they get used to that, and they associate the cues in that particular place with the cocaine. And so when they develop a dependence, if you put them back in that environment, but you don't inject cocaine, you inject vehicle or nothing, the animals will preferentially go to that place because they associate it with the cocaine. And all of these have shown some predictive value to human addiction. Alcohol models are fairly simple. Alcohol can simply, obviously, be dissolved within the water that's given to animals, and so that can be quantified as the animals begin to choose, let's say, between two different bottles. One has pure water, one has alcohol in it, and the animals eventually develop dependence where they preferentially go for the alcohol, even when they're very thirsty and have been deprived of water for a period of time. And then opiates are somewhat similar to cocaine, particularly with respect to conditioned place preference, which is used quite a bit for opiate addiction, as well as some self-administration. So that is how hypotheses are tested, how drugs are tested. Now, the anatomy of addiction, which is the most relevant to the neurosurgeon, is summarized here. It is obviously more complex than this one cartoon, but the regions that are most interesting to the neurosurgeon are primarily the mesolimbic dopamine systems, which go from the ventral tegmental area to the nucleus accumbens. Now, the ventral tegmental area is a mesolimbic dopamine system that is just medial to the substantia nigra, pars compacta, which is the dopaminergic system that is part of the motor circuitry that goes to the dorsal striatum that most functional neurosurgeons are familiar with, because that is what degenerates in Parkinson's disease. The ventral tegmental area, which is another dopaminergic population in the midbrain, does not degenerate in Parkinson's disease. And in fact, a lot of research is ongoing to try to understand why the VTA neurons are more resistant to degeneration than the substantia nigra neurons in Parkinson's disease. But the VTA dopaminergic neurons seem to subserve the reward and ultimately the addiction systems by sending dopamine to the nucleus accumbens and ventral striatum. And so it is well established that dopamine is a major mediator of reward and ultimately addiction. And if you look at the chemistry of addiction, you can see that the various drugs of abuse work on these systems at different nodes. So, for example, nicotine works on the VTA neurons and can be involved in depolarization, etc. Cocaine and amphetamine can influence release of dopamine at the nucleus accumbens. And then opiates influence the depolarization of inhibitory neurons that regulate the VTA that are seen here in red. There's another area that's been of great interest in recent years, which is called the lateral habenula. And those are glutamatergic neurons that project to the inhibitory neurons of the brainstem that regulate the VTA. So by activating those inhibitory neurons, the lateral habenula inhibits the VTA and regulates it that way. So these are all potential targets for the neurosurgeon. Now, there's a very limited literature on human neurosurgery for addiction, but all of these things, all of these areas are potential targets and have been postulated as areas that can be targets for deep brain stimulation or more advanced biological therapies. And so finally, the literature on what has been done in humans begins, roughly 50 to 60 years ago with radiofrequency lesioning or other types of lesioning of circuits that to try to influence addiction. So various limbic white matter structures, so-called limbic leucotomy, cingulotomy, which many neurosurgeons are aware of because of its involvement with other disorders. People have done cingulotomy as well as cingulate stimulation for obsessive compulsive disorder, for severe pain. What limbic leucotomy and cingulotomy mostly do is create a decrease in affect, a decrease in interest overall in things. So it can treat addiction, but it's doing it usually in a way that doesn't really normalize behavior, but substitutes apathy to some degree for addiction, which is the problem. That's even true with many drugs, that many drugs that can be helpful for addiction promote some element of depression because they can often be two sides of the same coin. There have been lesions in the ventromedial nucleus of the hypothalamus. This is an area that I had done some research on early in my PhD many years ago. And the problem with that is that the VMN is a very multifactorial structure that is involved not only potentially in regulating satisfaction and reward, but also is a key regulator of sexual behavior, of body weight, and of other things. And so, not surprisingly, there were all kinds of complications when one lesions the VMN of the hypothalamus. And then there's nucleus accumbens lesions, which actually have been of interest even more recently, and have been these have been done even with stereotactic radiosurgery. So that is something that's potentially feasible in an addiction patient. But again, given the importance of this area to depression and other things, it is not entirely clear that lesioning the nucleus accumbens as a whole will be without cost, even if it was effective for addiction. Now, part of the techniques that I mentioned earlier are the optogenetics and others have shown that subregions of the nucleus accumbens, such as the shell versus the core, can influence very particular aspects of addictive behaviors as well as depressive behaviors. And so being able to target these areas more specifically may provide greater benefit than lesioning the structure as a whole. And then, of course, there's neuromodulation. So vagus nerve stimulation is actually FDA approved for depression, and not surprisingly, there have been some animal studies looking at vagus nerve stimulation with addiction. The neurobiological theories behind this are a bit are not entirely clear, but similar to depression. Likely, this is something that is working through the hypothalamus and maybe influencing these dopaminergic circuits if it has promise. And then, of course, there's deep brain stimulation, and that has been tried in some case reports in very small series for both alcohol addiction as well as other types of addiction. And that is something that is of great interest because of the potential to not simply lesion areas like the nucleus accumbens or possibly the lateral habenula, but to actually modulate them, which would be more preferable. And then finally, in the future, many of the experimental therapies that neurosurgeons have been intimately involved in developing and in using in human experimental subjects, such as gene therapies, for example, will allow us to take many of the reagents that are being used experimentally in animals right now and potentially translate them directly into humans, not simply as research agents, but as actual therapeutics, because so much gene therapy is being done in the human brain right now for diseases such as Parkinson's disease, for genetic diseases, now even for things like Alzheimer's disease and others. And there are a lot of other things that are on the horizon to be tested in patients soon, that it would be surprising if gene therapies did not wind up being used in humans fairly soon for addiction as well. And that creates the opportunity for a much more cellularly specific and even molecularly specific therapy that could really change the opportunities for addiction treatment surgically. And finally, MR-guided focused ultrasound, which has led a resurgence in interest in lesioning the brain because it is a non-invasive method for lesioning, could be used to lesion some of these structures that had been lesioned historically non-invasively. But also, MR-guided focused ultrasound can be used to open the blood-brain barrier, and there are many human trials going on right now in brain tumor therapeutics and in Alzheimer's disease, where the blood-brain barrier is being opened with focused ultrasound to try to treat those disorders. And again, this could be used to deliver therapeutics such as gene therapies or cell therapies or antibodies or other things that could modulate the brain non-invasively. And so the neurosurgeon of the future will have a lot of tools at their disposal to try to treat a disease such as addiction, which has an enormous unmet need, an enormous population that the neurosurgeon might be able to help in a way that could take advantage of our skills and our ability to understand anatomic specificity and deliver payloads to particular brain regions in order to improve this devastating disorder. Thank you very much.